Hello students, welcome back to Rainbow class. I'm teacher Theophil, teacher of biology of senior 5. So we are on the unity number 5 called the cell and nuclear division. So we have seen the previous lessons as we have seen haploid, diploid conditions. We have seen the cell cycle, and then now we are going to see the process of mitosis. Dear students, who can tell us the meaning of mitosis? So we have seen this mitosis as division of nucleus, then to produce two daughter nuclei identical to their origin cell. Yes, you understand it. And then this mitosis is also one of cycle or one phase of a cell cycle that we have seen. So this mitosis from one cell to produce two daughter cells, it passes through 4 phases. The first phase is called the prophase. The second phase is called the metaphase. The next phase or third phase is called the anaphase. The last one is called the telophase. Dear students, follow me then to see the events that take place in each phase. Let us start. First phase. Also, before we start, also in the first phase, let us see also the structure also start by the interphase. You remember we have seen the cell cycle contain interphase, mitotic phase, and cytokinesis. So here, this diagram shows different events take place during the mitosis. Here, but before enter to the mitosis, it starts by interphase. Then also here, this structure, it shows the G2 or the second growth in interphase, means that the cell growth. Then the centrosomes with the centriole pairs exist in this interphase in G2 or second growth. Then also, this nucleus also exists. The nuclear envelope also exists. The plasma membrane also exists. And also, chromation or duplicated of the chromosome also exists. And then after the G2 of interphase, it is followed by the next phase called the mitosis phase or the mitosis. So, then here there are different phases of mitosis. The first phase is called the prophase. Here the structure that's summarizing the event, but we will see in deep different characteristics shown by each phase. So first phase called the prophase. Here there is mitotic, or here there is what we call the spindle or the spinal fibers. Then also there is a sister or the aster. Then also here you found the centriole. Here the centriole also they contain the hair-like structure called the aster. And then also we see the chromosome consisting of 2 sister chromatids in this prophase. And also these 2 sister chromatids are joined together by a centromere. This for prophase. The next also, before we enter to from the prophase to the metaphase, there is also the preparation phase of metaphase called the prometaphase. In this prometaphase, there is a fragment of nuclear envelope. Here is a fragment of nuclear envelope. Means that nucleus that you found in the prophase, they start to disappear. Means that here is different fragment of this nuclear envelope or nuclear membrane. It starts disappear for this prometaphase, preparation of metaphase. And also there is what we call non-kinetochore microtubule that we found here. And also kinetochore microtubules also exist, but also there is what we call non-kinetochore microtubules. Then it is followed by the next called the metaphase. This is the preparation phase of metaphase, then metaphase. For this metaphase, here all the chromosomes are lined up to equator. Means that here in the metaphase, in an homologous chromosome pair up of homologous chromosome. means that here in this mitosis, you found the non-homologous chromosome. Then line up to equator, or this equator, then also you found, or is also called the metaphase plate. Equator or line up is also called the metaphase plate. All the chromosomes are lined up to the metaphase plate or equator. Then also found the centrosomes at one spindle pore, and the next also is found. Then also you found the spindle fiber. These are the spindle fibers attached on the centromere of each chromosome. Then next also is called the anaphase. After metaphase, there is the anaphase. Anaphase, it is characterized by the splitting of chromosome into chromatids. Here the chromosomes, the sister chromatids, are split up to the centromere by spindle fibers. They produce daughter chromosome that reproduce daughter chromosome. And then the next one, or the last one, is what we call the telophase. The telophase also, it is followed by the cytokinesis. In this telophase, there is also the form of nuclear membrane. Nuclear membrane also starts to appear or reform. There is a reform of the nuclear membrane in each nucleus. Also, nucleus, and also here there is what called the chromosome, the next chromosome. And then, these are the simple summary of this mitosis phase. So, let's see the characteristics of each phase. The first phase, prophase. And then the prophase, the chromosome fibers become more tightly coherent. Means that here the chromosome fibers become more tightly coherent together. The next characteristic of this prophase, as you've seen in the structure, the chromatid also condense into discrete chromosome. The next, each chromosome can be seen to consist of two sister chromatids, as we have seen in the prophase. The nucleolus shrink, eventually disappear in most species. When you say shrink, the size decrease. Their size decrease, and maybe sometime or some species, these nucleolus disappear. Then two pair of centrioles are seen, very seen. Then this mitotic spindle assembly outside of the nucleus. These are also the characteristics of prophase. The next also, there is what called radio arrays of shorter microtubule are called the aster. We have seen the aster on the previous structure. We have seen the aster. Aster, yes, we have seen the aster. This is what called aster, hair-like structure that found around the centriole is called the aster. And there also the aster are formed in the prophase of mitosis. The aster also is formed radio array of shorter microtubule called the asters. These around the centriole. And then in most animal cells, the centriole are a focal point for the spindle assembly. And then also for the higher plants, do not have a centriole. This exception. For the animals, you found a centriole, but higher plants do not have a centriole. But they do have mitotic spindle. So then, these are the characteristics for the first phase called the prophase. Dear students, if you understand well, who can remind us one characteristic about the prophase of mitosis? So we have seen those. Then, let me continue for the metaphase. So this metaphase, the main event for this metaphase, the chromosomes, non-homologous chromosomes, line up at the metaphase plate or equator. But also there are different other characteristics that shown in this phase. The next phase also, for this metaphase, the next characteristic is the centrosomes are now at the opposite pole of the cell. Means the centrosomes, we have seen the structure, the centrosomes are different opposite pole. This is what called metaphase. Here there are centrosomes on each pole. The one pole, they contain centrosome, and another pole contain centrosome. Centrosome is also a characteristic of metaphase. So the next also, after these centrosomes, Also, the kinetocoa microtubules form two pores, orient the chromosome in such a way that their centromeres become aligned with the metaphase plate or equator. Then also, there is another or third phase of mitosis called the anaphase. The anaphase also, we see that the chromosomes separate. Single chromosome will produce chromatids, means the sister chromatids separate at centromere. In this phase, here once the centromere are attached to two sister chromatids, they start separating and they move toward the opposite pole in this anaphase. But also, here consequently the centromere sister chromatids separate and give rise to daughter chromosome at the end on the telophase. So then also, the separation continue until two pores of the cell have equivalent and completely chromosome. Means single chromosome separate into two in the centromere to produce two chromatids that will form chromosome later. And then the last phase, this is the fourth phase to the 5, is the fourth phase called the telophase. The telophase is the last phase of mitosis, where the two sets of chromosome or two sets of daughter chromosome are assembled into two globes at the opposite end of the cell. Then the chromosomes begin to anechoid, start again anechoid, assume the elongated state of characters of interphase as we've seen, the characters of interphase. Also, the nuclear envelope starts forming, means that for the prophase, the nuclear envelope disappears. But now, on the last phase, also, in each daughter cell formed, the nuclear envelope starts to reappear or form. The spindle fibers or spindle microtubule disappear, and the nucleolus or nucleoli form again. Where for the prophase, the nucleolus disappear, and also nuclear envelope disappear. But now, on the last phase of telophase, these spindle microtubule disappear, where the nucleolus and the nucleoli or nucleoli plura reform. Then, at this point, the nuclear division is complete. Means single cell produce two daughter cells identically in genetic materials. Then has two identical nuclei. Means that their genetic material are the same. So here also, let us see also the cytokinesis. After this telophase is what called the cytokinesis. What's the cytokinesis, dear students? We have seen the cytokinesis. In the previous, we have seen the cytokinesis. The cytokinesis is a division of cytoplasm. Then this cytokinesis. In the cytokinesis, there are different characteristics. It shows different characteristics. The first cytokinesis has the meaning of division of cytoplasm. This is the division of cytoplasm. I think that you understand it. Dear students, also for this cytokinesis, there is a complementarize the two new nuclei into separate daughter cell. Means each nucleus have also the nuclear membrane. They form a new cell, a completing mitosis and cell division. So in animals, different from the plants. In animal cell, cytokinesis is characterized by the constriction in the middle of the cell. The constriction continues until two daughter cells are produced. Dear students, here, in animals, cytokinesis is different from the plant. In animals, it is cytokinesis characterized by the constriction in the middle of the cell. The constriction continues until daughter cells are produced. While in the plant, new cell membrane, cell wall are assembled between the two nuclei to form a cell plate. And then also, the cell wall materials cut each side of the plate and result into two progeny cells. So, but also, let's see some summary of events in the interface and also mitotic cell division. Then let us see by the comparable table showing the event for each interphase, mitosis, and cytokinesis. Here is a table that's showing only one characteristic that's summarizing these different events in interphase, mitosis, and also cytokinesis. Let us see the mitotic phase or M phase. Then the prophase. There is condensation of chromosome in nucleus. Condensation of chromosome in nucleus is the summary of the prophase. Metaphase. The metaphase, the chromosome align at metaphase plate or equator. I think that you remember, dear students. So, then also anaphase. During the anaphase, the chromosomes are separated into two clusters at the two poles. The telophase and the cytokinesis, the two clusters of the chromosomes have reached the pole and formed the two daughter cells. And then also in the cytokinesis, there is separation or cytoplasm has taken place. And then in interphase, you remember it contains G0, G1, S, and G2. So for G1, G0, you remember the cell is in resting, no activity performed. Then for G1, the cell prepares itself for DNA replication. Means the main event for this G1 or first growth is DNA replication. Where synthesis phase, DNA replication takes place, this DNA takes place. While for G1 is the preparation of DNA replication. While the synthesis phase, DNA replication takes place. So then also the second growth or G2, then the cell resume growth before the cell enter the next phase. It means mitotic phase. And then these are the like a summary of the event interphase, mitotic phase, and cytokinesis. Dear students, I think that you like this course. This lesson was interesting. If it's interesting, I think that, dear student, is interesting. Please subscribe and comment. And then we will meet next session. After this, we will see the importance of mitosis. Dear students, thank you for your attention and kindly then see you next time. Thank you.